Hello students. Today we are going to discuss about pyridoxine. It is also vitamin B6 and it is also called as antidermatitis factor. If we see the structure of it, it is in three forms. One is pyridoxamine, pyridoxal and pyridoxine. All are pyridine derivatives. If we see their active forms, they will be presenting in phosphate esters. Pyridoxamine in pyridoxal phosphate and pyridoxal in pyridoxal phosphate. This reaction will happen by phosphorylation and by the enzyme pyridoxal kinase using ATP. If we see their metabolic role, in its active form, B6 will act as coenzyme in proteins by shift space linking with lysine residue, and it also helps in amino acid metabolism. We will see different reactions how B6 will mediate through PLP, which is pyridoxal phosphate. One is your transamination reaction. All the amino acids undergo transamination reaction except few things like lysine and threonine. And next thing is decarboxylation reaction of amino acids. 5-hydroxytryptophan converting into serotonin, glutamate converting into gamma aminobutyric acid, dopa getting converted into dopamine, and histidine getting converted into histamine. All these four reactions are decarboxylation reactions where the enzyme is decarboxylase. The first three are neurotransmitters, where glutamate is inhibitory neurotransmitter. Because of the deficiency of B6, there will be a sorry, not glutamate, it is GABA is the inhibitory neurotransmitter. If there is any deficiency in B6, there will be a decreased production of GABA, which will lead to seizures. We will see it later. Now, third reactions are non-oxidative deamination of hydroxyamino acids, where serine is getting converted into pyruvate and threonine is getting converted into alpha ketobutyrate. In this reaction, ammonia is going out and the enzyme is dehydratase. Now, in the fourth reaction, cleavage of threonine, the fourth reaction, cleavage of threonine into glycine and acetaldehyde. Fifth reaction is interconversion of serine and glycine, and next is synthesis of alpha amino beta keto adipic acid and in ALA synthase. This is the first step of heme synthesis where ALA synthetase uses PLP, which is pyridoxal phosphate. With the help of that, succinyl CoA and glycine are getting converted into ALA. Now, during the tryptophan metabolism, in tryptophan metabolism, we have three pathways. One is kynurenine pathway, one is indole pathway, and another thing is serotonin pathway. In the kynurenine pathway, during the conversion of hydroxykynurenine to 3-hydroxyanthranilic acid, B6 with the help of enzyme kynurenase with the help of coenzyme B6, this reaction is happening. What happens if there is any deficiency of B6? This kynurenine will get converted into xanthiniuric acid, which will be excreted from the urine in the form of xanthinuria. Now, in the eighth reaction, it is transsulfuration or desulfuration reaction, where you will see this thing in the methionine metabolism. And in muscle phosphorylase during glycogenolysis. And in sphingomyelin synthesis, coenzyme A synthesis and amino acid transport into the cell. These are all the reactions which require vitamin B6. Now, we will see the deficiency manifestations. Usually, deficiency by vitamin B6 is uncommon. If at all present, these will be the manifestations. Neurological symptoms, which is mainly because of the decreased GABA and even decreased synthesis of sphingomyelin, which is required for the neural sheet. It will lead to neuritis and seizures. And dermatological symptoms, since tryptophan getting converted into niacin requires B6, any deficiency of B6 will lead to pellagra like symptoms and hematological manifestations. Since the first step of ALA synthase, which requires B6 in the heme synthesis, it is not happening. B6 deficiency will lead to sideroblastic anemia. And xanthinuria, as we explained previously. Hyperoxaluria, alanine and glyoxylate transamination requires B6. If B6 is deficient, glyoxylate cannot be getting converted into alanine, and this glyoxylate will be getting converted into oxalate, which will be excreted in the form of hyperoxaluria. Now, because of this oxalate, there is a risk of renal stone formation. Now, homocystinuria. During the metabolism of methionine, cystathionase enzyme requires vitamin B6 as a coenzyme. Any deficiency will lead to the accumulation of homocysteine, which will be excreted as homocystinuria, which is a risk factor for coronary artery disease. Now, how will you assess pyridoxine deficiency? One is we have tryptophan load test. Tryptophan load test is like giving the tryptophan to a patient and you are assessing xanthiniuric acid. If there is any deficiency of vitamin B6, tryptophan metabolism cannot happen properly. There is a increased production of xanthiniuric acid. So we will assess xanthiniuric acid so that we can come to know if there is any pyridoxine deficiency or not. SGOT activity in RBC. All the transamination reactions require B6 so that we can find out if there is any deficiency of pyridoxine, and directly finding the plasma B6 levels. If they are less than 20 nanomole per liter, then there is a pyridoxine deficiency. The required daily allowance of pyridoxine in adults is 1.5 to 2 milligrams per day, and in pregnancy and lactation it is 2.5 milligrams per day. The dietary source are whole grain cereals, unpolished rice, legumes, green leafy vegetables, yeast, egg, meat and milk. Thank you.